into the introduction. Before I begin, so thanks to Jana Jofan, who is now professor at Boston University and Bar-Ilan University, Marco Punta, who's at the ICR in London, Avner Schlesinger, who's professor at Mount Sinai, Karl Brzezinski is a group leader at the Genome Institute in New York City. Oh, there was somebody else missing. Protein function. Life begins how? What's the molecule that begins life? Is it trivial or? So I really need your help to get a feeling for what is trivial, what is a question that you all don't know, or what you all know. And I look into the same faces somehow. For me, it's very difficult to distinguish the way you look at me. It's not trivial, is it? No, okay. So if it's not trivial, then what is it? If it's a fun guess. How do you imagine? So somehow life starts with some molecule, right? It's not iron, right? It's not oxygen. This must be something that sort of has a propensity of storing some information, repeating that information, and doing all kinds of things. So what is that?
DNA and protein.
DNA, do you believe life starts with DNA and proteins? You're not that far off, but
Only DNA.
No, actually not, because DNA has one thing that is majorly missing. It cannot reproduce itself easily without an environment. I mean, in a cell it can, but so before you think cell, if you think a piece of DNA, that will not reproduce itself. Navid?
I wanted to say RNA, but it can't reproduce itself.
Yes, it can.
It can.
Yes, and in fact, that is the candidate. And it goes back to Manfred Eigen, who got a Nobel Prize for that, Walter Gilbert, Tom Cech is sort of a more modern person to make this point. In this universe, somehow the RNA that can in fact reproduce is the beginning. It sort of creates DNA and gets into the cycle that we know today. Now, in this cycle, RNA has some enzymatic activity. But if you think through, and this goes back to Manfred Eigen here, it goes back to the question, if we sort of have this original soup in which life develops, what kind of chemistry do we need? So essentially he went through all the chemical pathways to chemical processes that you would need to have RNA reproduce itself and to actually sort of begin to do that. And they stumbled over the fact that some things, some enzymatic activity, some reactions cannot be handled by RNA alone. In fact, for those, the argument was, what you need is peptides, proteins, amino acids. And that, in fact, is now the point in the animation of my slides here is strange. Well, Andrei Lukács comes into the game, and his idea, or many people now believe, that RNA was at the beginning. It found some amino acids, some peptides. It built something that helps it create life. And these some things it found, today's proteins, then if that is sort of right, then in today's proteins you should still see that history. For instance, in nature we have more than 20 amino acids. Why were those 20 chosen? Maybe because those 20 have exactly to do the kind of chemistry that RNA alone couldn't do. Maybe those 20 are enriched in their propensity to bind RNA. And that, in fact, is what Lukács and his group have demonstrated. And what they also do, essentially, they go through the history of folds. They try to see, so particular RNA binding motifs or particular fold motifs. Can you see over the history of the evolution that the core is the same, and does that point to an original peptide, the core of a fold, that in fact could be the RNA binding, or the one that does a particular enzymatic activity, and that could be sort of this precursor of a long protein, a short peptide that does some chemistry. And that's, in fact, they sort of collect examples where the story checks out like that. Or it's not they collect, they have been collecting, so he's the director of the Max Planck Institute in Tübingen since now over 20 years, and every year they are collecting new evidence, and over these 20 years they have strung up a story that is rather convincing. It's not causal, so they cannot prove it, right? It's just they can collect a lot of evidence that points into the direction that this, in fact, is a very good explanation. Life began with RNA that picked a particular set of amino acids, and those particular set of amino acids led to what we see today as the 20 amino acids of life. Today's alphabet is slightly different. DNA is the long-term storage. RNA is short-lived. So it created a material that sort of can keep information in a more reliable way, DNA. And in some sense, we often see here in the central dogma of molecular biology, the information flows from DNA to RNA to protein. That clearly is true in an information flow kind of sense. RNA as an intermediate step is not entirely true. I'll get back to that in a moment. In fact, that is the dent in this picture here. The intermediate, so clearly ribosomes can produce proteins without proteins, that we know already. So there's a lot of RNA in ribosomes. So we know that not all the machinery of life is proteins. Clearly there's RNA. And we know, most of you have heard about short RNA, RNAis. Those are short pieces that decide how proteins are expressed, that decide how things are orchestrated in the network of the protein machinery. They are regulators. So these are pieces of RNA that clearly regulate a lot in the cell. So by that token, they're not just temporary intermediate step. They carry information. And they have activity, in the sense activity not that you can measure an activity, but they do something, right? They knock out, they block a binding site, they activate a protein, they activate an expression. But beyond that, all of these here, typically we would argue, we are talking about something that is sub 50 nucleotides. Much less than 50 nucleotides. In fact, many of these here are just a handful or 2 handfuls of nucleotides. So for most of those, they are below 20 nucleotides. When I say most, I mean 99.9% or something like that. Okay, now are there longer RNAs? Are there things that are like 100 residues, meaning 300 nucleotides? Or something like that in RNA, and that sort of have an activity. Do you know any of these? Have you heard of those? Yes?
Maybe ribosomal?
In ribosomes, for instance, we have several examples of exactly that. Yes. That's one example. Anything else? So ribosome is a perfect example.
I don't know, I think long non-coding RNAs are defined as more than 100 nucleotides, so you have several of those.
There are several of those. When you say several, what do you mean? 1, 2, 3, 4, 5? 17? 42?
I think less than
137?
Less than protein coding was, but not that much less.
Exactly. And that in fact was published at this point 13 years ago. The first view on this was published by a consortium in Japan. The Riken consortium, so this is the so-called Phantom-3 consortium, functional annotation of mouse, one of those things with 143 authors, and essentially overnight what they showed here is that the number of not protein coding transcripts here is in the same ballpark of the number of proteins they saw in mice. So for that particular data set they had about 16,000 proteins in mice. So we believe that some of us believe that mouse has about 20,000 proteins, but the data set, they did not have them all in their data set, right? So the data set they had about 16,000, and the non-coding ones, non-protein coding ones, is also 16,000. So it's exactly sort of what you said. It's the same ballpark order of magnitude. We're not talking just, the 20 that are on the ribosome and maybe 20 else. We're talking 16,000. Okay? And we still don't know at this point, so 13 years after this publication, how much of this number here is due to diversity, so meaning that we really need different functions for different families. So over the 13 years, people have described many of these functions, but do we need them all? We still have no idea. Okay, this number continues to actually stun us. So it is actually not true that RNA is only an intermediate step. It's a universe of non-coding RNA. And it may actually be time to somehow get rid of this, bury this dogma is what I write there. But for the continuation of the lecture, we will anyway focus on the machinery, the proteins. And that brings us to the question, what is protein function? how would you begin to answer that question, what is protein function? Anybody has an intuition? Throw at me what is protein function. How would you define protein function? Any idea? Any suggestion? Can you define a protein function? Yeah?
I mean, essentially it's just function carried out by proteins, but I mean there are many different functions, many different kinds of proteins. I mean you have enzymatic activity,
Yeah.
for example. You have structure proteins, you have signaling proteins, you have all kinds of different functions, so I think it's difficult to actually define it in just one
Word.
sentence or word, yeah.
So let's just take, I'm going to take your definition in a slightly rephrased way. There are different types of function. So protein function, there is no one single definition for that. There are different type of functions, different type of proteins. Well, you talked about enzymes, you talked about structural proteins, and we will get back to that. In fact, this is very much the operational definition that will guide us through the rest of the lecture. I will give you a little bit more, but you will not come out from this lecture being much wiser than this. But before I shock you with this, let's shock you a little bit more. Let's begin with a much simpler question. What is a protein? What's a protein?
Sequence of DNA.
Actually, it's a sequence of amino acids. Yeah? Fair enough. Yes, that I believe. And in fact, unfortunately, Issa gives a definition that is not far away from what you will end up with today. But try to do better than that.
The sequence has a specific structure.
Ah, that's an interesting idea. So you define a protein essentially. So let's begin with the first one. Issa says it's a sequence of amino acids. And that's what we have here, right? Now, you say that in fact proteins are only the subset of all the possible sequences of amino acids that adopt a unique three-dimensional structure. And I find that's a totally cool idea. I really find it very cool because I never thought about that. That is a very original definition, and it nails a point, because we will never observe a protein that does not fit that definition. So this is a totally cool idea. And by that definition, I always think about the universe of all possible protein sequences, and I think about the many that don't exist. And you just don't call them proteins. And in that definition is, actually, I sort of like that idea. Okay, anyway, so we have a sequence. Now we take this sequence from Anfinsen, who got a Nobel Prize for that. We essentially know that we put that sequence into water. Mostly what we get is a unique three-dimensional structure and a unique function that comes from that structure. Here are some of these structures from the first paper of Christina Orengo-Carth, Christina Orengo and Janet Thornton. Just a multitude of different protein structures. Here are some more. In particular, we have membrane proteins here, channels that let ions through. Here, something that related to rheumatitis, a disease here, the GP120 protein related to HIV for the virus to get in. Typically, any first step of sequence comparison, we compare a particular protein sequence against another protein sequence. And now back to the question. Most people would tend to argue, well, a protein is something that comes from a genome. A protein comes from a gene, is what many people will answer. And then I will ask, so what is a gene? But the typical answer to that one is sort of pointing to some region on the genome that in some sense, whether it's transcribed differently or not, spliced differently or not, but that is somehow co-expressed, right? You have a region on the genome, and that entire region is, or some part of that region at least, so if maybe it is like this one here, I to J, position I to J, and maybe this one here is cut out, maybe this one is cut out, maybe they're spliced together like this, in that order of things, all of that, but it's all from some local region. All that ends up being the protein is in fact coming from that region between I and J. Is everybody happy with that definition of a protein? Whoever is happy with that definition of a protein, whoever has taken my course before, the correct answer of course is no, although you don't know yet why no. Here's an example for where this definition goes totally wrong. The ATPase synthase is this entire block here that we typically again write as one long sequence, a very long one here, and here you see subunits A, B, A, B, A, B, so there are several that come together of the same sequence of the same protein, and they aggregate in particular ways. In fact, they rotate in the cell, right? And this motion of rotation is happening in the middle of it, in the needle of it. They're essentially partially done by two helices winding around each other that turn around, and that turns this entire mushroom around in the cell. And creates energy. Anyway, so the main point is we perceive this as one protein. And that protein again we can describe the functions in a variety of situations. It moves some ions around. We can even show how many pumps this is doing in a certain time scale. We can really macroscopically measure it. The tricky bit about this here is that the sequence is not coming from such a region. Actually, it's coming from different chromosomes. We have an example here. We have one protein that is coming together as a protein sequence from different chromosomes. Okay, it's completely against this sort of linear idea, a piece of DNA is a protein, yes?
So in this case you would consider the complete ATPase as one protein?
Yes.
Because, I mean...
So it has subunits. It has subunits. So that is true. But there are parts that would... so there are different parts here.
Because some people distinguish between like the single protein and protein complex.
That's the point. This is not a complex. That's exactly it. So the ATPase synthase is not a protein interaction. Well, at least it's not commonly thought of, because these things do not. So the way we typically think about protein-protein interaction is we have two things that come together and could possibly bind to something else, right? This is one unit. It always is. It's just behaving in any way that we can measure like a single gene. It's behaving, and that ultimately is the reason why this is considered a gene, right? With, again, there are subunits, they have to come together, there's a variety of issues. But again, so you have this very often also for things that are collinear, so that come together from the same I and J region. This is just one example. So all I'm trying to point out here is that the story is not that simple. Even the clear definition, and by the way, this slide should have been before, so sequence comparisons have to do with finding out from the comparison of the linear sequence that these two here are related, that in fact the top one is the cytochrome P450 for peppermint, while this one here is for spearmint. And that brings us back again, can we define function? So here is one example of this is not quite a definition of function, it's a description of function. And it comes from Protopedia. If you don't know it, it's a very interesting resource to look at proteins. So you get a structure. You see, so Protopedia is strongly driven by structure. I believe that actually all entries, maybe Protopedia has something about disorder we get to later, but mostly it's driven by proteins for which you know structures. And then you have a functional description. Okay? So let's take this functional description, the abstract, and sort of formalize it a little bit. So one thing that you see is a number. And that number will be with us for several lectures, the EC number, the enzyme nomenclature. The EC number has, in fact, a hierarchy on 4 levels. The first number is sort of the lowest level distinguisher. So this is, for instance, in this particular case, an oxyreductase. So that is the major reaction type it catalyzes. While the 4th digit here is the full detail of the reaction, and the digits in between show something about the alternative ligands or substrates for an enzyme. Now we get to something else that is in this abstract here that is not at all described by this enzymatic activity function, which is, it is the 4th step in metabolism of fructose. So it's breakdown of sugar. That means there's a process, there's a pathway, there's a set of things. In this set of things, the blue ones are our proteins here. This is the entire fructose pathway from the KEGG database. Kyoto is what the K stands for. And it is one of those elements in this entire pathway. Okay? It's one step in a larger process. So, back to the point, being part of a pathway is also an aspect of function. So now we have, this is an enzyme, and so you said there are structure, there are enzyme, but we have actually a different aspect here. It's a part of a pathway, right? Because it's an enzyme, but it could be actually, maybe there's a structural protein in here, right? It could be part of that pathway, whether it's a structural protein or an enzyme. Okay? So it's a different aspect of what it does. It participates in some process. It's like whether I classify you according to whether you're a woman or a man, and then whether you are a student at TUM. Those are two completely different classification systems, and they both are equally true, and they describe different aspects of who you are or where you are. Okay, ultimately, when this is coming back to, you said there are different types of proteins that have different types of function. The story is more complicated. Whether you ask or you consider as function how a particular reaction is done by an enzyme, how a particular bond is cut, you could consider that as the function, and that would be sort of the chemical or biochemical view. Or you could consider it as the aspect of function, whether it's a transferase or an oxidoreductase. So more what type of generic reaction this is doing. You could say that this particular protein is part of the cell cycle. So whenever you have a budding of two cells, this sets on some reaction, right? You could say how that sets on that cell cycle event, or how that sets on any sort of transfer event, any pathway event, that's its function. You could simply say when it regulates. So particular types of cells are triggered by this event. So that's regulatory. It's a developmental question. You could say its function is that it has something to do with a disease, or does some knockout of a disease or whatever. Or you could actually look at the genetic level and say it's dominant or recessive. It is doing something or not. And depending on what you study, all of this is equally good as a definition of protein function. And all of these are very different things. It's not only the different thing that you alluded to when saying structural protein or an enzyme. It's much more. It's really, even on the level of the enzyme, what you're looking at. What aspect of the enzyme you're looking at. Depends on what you want to study. And none of that is really a better answer. There's no better answer here. None of those is more right than anything else. So you can try to address that by saying function operationally is anything that happens to or through a protein. That sort of covers everything. It is good. The beauty of this definition, it's right. The sort of bad side of it, or the ugliness of it, is it doesn't say anything. But anyway, so let's call it an operational definition. In this operational definition, again, and that's a slide from Ana Luisa Arrudo, where we look at leptins, and we simply look at the same protein in different types of cells doing very different things. So here it has an increased heart rate, and here it regulates appetite. You may completely say, for me, my heart rate or my appetite, I would, in principle, sort of assume that these are different functions. It's the same molecule, just in a different cellular environment, right? And so here more details. Here's one of those things that I like a lot. Tales is a book by William Geritzen, and here's in fact one example. Tales is just a book full of stories about what protein function is. This is one particular story. Here is another one. There are many of these, and in fact she has collected them for Swissprot over many years. I believe they are freely available online, and some of these stories are really remarkable, and it just tells you a lot about the diversity of protein function, about the complication of defining function. Again, this is in P53, one particular transcription factor, and there's a small region of binding sites that really have very different impacts on different sites. Here we have an example where the opposite is the case. We have one particular variant, so there is an E, amino acid E, mixed up with a V, so changed against a V. It says at position 6. It happens to be at position 7 in that protein. I'm giving you that detail because these numbers, whenever you see these numbers, be wary. Some people cut the first M out of the sequence because everybody knows that every sequence starts with an M. So that's why they count, simply the ones who annotated it initially never counted the M, because in those days every sequence started with an M, so they didn't count the M. In reality, this is position 7. Whatever, there's a single change. A single amino acid is changed. For everyone who carries that change, the hemoglobin will actually aggregate in cells that have a sickle shape. These images are not good, I'm sorry for that. But it will essentially cause sickle cell anemia, and that is a bad disease. Again, it's a single variant, a single amino acid variant in your entire genome that causes the formation of these fibrils that are not good in the red blood cells. Okay, so again, sequence determines structure, structure determines function. But what you have to understand is that structure is a scaffold for function, and that this scaffold essentially works only as a scaffold that allows motion. Function happens through motion. Function is binding. A binding event means always that something comes close to it. And I'm going to show you some movies here that show this reality. We can in fact do this. So last semester we did not quite get to can we predict 3D structure from sequence completely, but we can in fact not predict 3D structure from sequence completely for a sequence for which I have no homolog in the database. We cannot predict from scratch through molecular dynamics, but what we can do through molecular dynamics is, if I have an experimental 3D structure of a protein, apply molecular dynamics to see where is motion. And that's what I'm going to show you on the next movies. So here's the first one, where substrate comes, some molecule that binds to the protein, and in this protein you see that there are regions that move less and regions that move more. In particular, the loop at the outside here, or the sort of helix, flubbering out helix there, is moving extremely much, and you sort of see that the helix is actively, or more or less actively, moving the substrate along. This is Brownian motion. In that sense, it's not quite active. It's not a directed movement, right? But it is sort of a helper movement. It's sort of creating, lowering the energy barrier, and essentially makes this molecule here ultimately go to the place where it is happiest, which is in fact the binding site.
Why are some parts green and some orange?
So the coloring, so if this is the color that you mean by green, so all of these count the substrate at some point, touch the substrate at some point. That's the reason for making them yellow. Is that the one you meant by green? Yeah. And then what other colors? So this is the ultimate binding site here, and this bluish thing is the substrate, and the substrate has the oxygens on top, marked just for the energetic behavior of this. Is that answering your question?
Yeah.
Here we have another example where again this is a different view of the same molecule. This is from above, this is from the side. It's a membrane protein. So the membrane helices are orthogonal to the transmembrane layer. And what you see here is essentially the way to prevent the ions from getting through. So you have essentially, this is a channel, let's these 3 green ions through. 3 are essentially in waiting. They cannot get through because this thing is wobbling there. And at some point this thing, it will actually wobble out, and the first ion will get through. I'm not entirely sure whether in the motion, yeah, so you saw the first one coming out, you begin to see that it gets out. I'm not entirely sure whether we get to the end of it. Maybe, and again he is now, no, we never got to the end of it. He is now in London. And you saw that this one moves much less on that level. So on that side, it moves much more. And now I'm going to show you an example of where David Shaw gives a brief, it's a cutout from a talk of David Shaw that David Shaw gave at Washington University. So David Shaw developed this special purpose machine called ANTON. I believe at the moment they are at ANTON 2. That is by far the best solution for doing molecular dynamics today in the world. So with that, we get to David
water
Shaw.
molecules, and in fact, it's a very snug fit. This is when it finds its binding site, if you consider the water as it's fitting very neatly in there, which is what the drug really wants to do, among other things. So here's another one. This is also binding to sarkinase. It's a drug called dasatinib, and it is an actual drug that's approved for leukemia. And it's known again where that binds, and this is another experiment Yiping did where he just threw it into the box, and it's harder than that, but put it in the box, and it fairly quickly, in this case, found the rough right area. It's going to pause here, and it's actually kind of interesting because, well, maybe it already did it. A little groove opened up here, and it got distracted by it for a second, but then eventually it goes to exactly where it's supposed to. And then this is a different kinase. It's the epidermal growth factor receptor, which is it's actually a family of 4 different proteins that are implicated in several different indications. But in this case, for this drug, the drug is approved for the treatment of breast cancer. It's called lapatinib, and I guess it's hidden right now. This is the last one I'll show, but the drug's going to search around again, and it's going to find the right place. And again, there are a few false places that it is before it finds the right one. One very interesting thing about this is, this isn't like a new finding, but is what's called induced fit. In the process of having the drug molecule fit into a protein target, it's not just that the drug itself moves around, it's also that the protein moves around, making space for it. And the induced fit part is when the drug actually gets there, you can actually change the shape of the protein locally. And many methods to computationally identify the drug binding site and look at how well it actually works as a drug assume a rigid protein. They might actually start with several different snapshots of the way the protein could be, and then what's called dock all of these little drug molecules into it, see where they can fit, not through the actual dynamics, but just kind of moving around like this in all the possible rotations. But there's some kinds of drug binding which probably occur in areas that aren't even pockets. They're not, usually these kind of drugs bind to little depressions, and they fit very nicely, and they have certain types of chemical interaction. But in some cases, we don't think they're there. This has been postulated. We're seeing some examples that I haven't shown you where it appears to create its own pocket where you couldn't have predicted where it was going to bind and then bind in there. But this is all very, very preliminary, intriguing from the viewpoint of drug design, but early stage. So switching to computer science for a sec, ANTON was designed by a classic.
And there we are. So we're not going deeper into the ANTON lecture. But the point that you already saw before is that the aspect of dynamics is essentially part of how the structure looks, is how it can move. So it's ingrained into the shape of the 3D structure. I can move, or here I can move, or there. And this movement, in fact, binding is an active event from both sides in some sense. And I believe this is something that we have to think about. Now, in all of this, again, a protein sequence adopts a unique 3-dimensional structure, and this unique 3-dimensional structure is not solid, it's movable in some parts, in the right way. And this is order, and this order is relevant for function. And now comes the big exception, disordered regions. Disordered regions are, in red here, you have a region that is such that if you could shine light and see it at 2 different time points, you would see different things, or different states, right? You would not see a consistent image. The moment it folds, in this particular case, they're not all like that, but in this particular case, it adopts something that looks like a regular helix here. Here again, a little bit like a helix. So now, upon binding, now you shine light on that, if you could. Then you would at any time point you would see a consistent signal. Okay, so you have a transition from disorder to order upon binding, and so that means we have some. In fact, there is some disorder, and there's a quote wrong here, so I quickly advance on that. But we have some disorder that is such that it will in fact not be induced to forming order. It will always remain disorder. So some reason for disorder is to always be disordered. And what we in fact see is that in eukaryotes we have much more disorder than in prokaryotes. And people have sort of linked the degree of disorder to complexity. So arguing that having a lot of disordered proteins or regions helps you to increase disorder, because if you have a region that is flexible, then you can bind to different substrates. So you can make that bind to two things that look different, and binding to two different ones means more than one, right? So in the lock key mechanism of folding, and David Shaw alluded to that one too, in the idea where solid 3D structure or rigid 3D structure is the keyhole. You come with a key, that's the substrate, and the key fits into the keyhole. In this image, the protein structure doesn't change, the key fits right in there. In this image, there's one key to one hole, right? And the advantage of disorder could be that you essentially would create different holes on the fly, or you could use different keys. It's like this tool that burglars use to get into different doors that preferably are not their own. Anyway, so there is some evidence for the truth of this. Let's get back to terminology and metrics. So are there metrics for proteins function? So again, for sequence we have many different matrices. We have E-values from BLAST, we have pairwise sequence identity, we have many other values. For comparing structures we have RMSDs. Again, we can compare local regions. We can just essentially look atom by atom, how do they differ to structures, right? It's very easy to define. Maybe RMSD is not the best solution, but it's very easy to write a solution. What about function? So let's get back to enzymes. For the case of the molecular function aspect for enzymes, we have again the 6 first classes, oxidoreductases, I said that before, and I believe I had an example for a transferase, ligases we had. So on the first level we have 6 different types, then we have many more classes. Now we can simply compute a distance between two enzymes according to this tree. That will be a metric. Okay? For enzymes we can do that. There are some limits in this, but essentially we can do that. By the way this nomenclature is done is by experts simply looking at what the biochemistry of this particular enzyme is and coming up. Any new enzyme is discovered, it's put into the classes that exist, or a new one is opened. So some numbers don't exist, and in fact there are people trying to discover all the enzymes in nature that have not been described yet. There is another way in which you today, and I will get back to that in a moment, use enzymes for industrial purposes. For instance, you could create a bacterium that eats up plastic or something like that. In fact, that does exist, and since you smile is even more weird, there are bacteria. So now I was about to say that naturally eat plastic and actually produce plastic. So biodegradable plastic is produced by bacteria that in fact produce plastic by themselves. Okay, this is sort of, if without people having put plastic into nature, they wouldn't exist. But it's not that somebody went there and taught that bacterium to produce plastic. So biodegradable plastic comes from bacteria that naturally produce plastic for whatever the reason in their local biotope in their environment is. Anyway, so for a lot of modern processes in food development and drug development, you actually use bioenzymes to speed those up. But it's also true that not all enzymes of the organisms that exist today are known. Okay, here is the attempt to do something like that for all proteins, so including the structural proteins. So something like that is called the gene ontology. The gene ontology has 3 different if you want, ontologies, or 3 different hierarchies, or 3 different classes. There are different words for that. And it all goes back to Michael Ashburner. Now, Michael Ashburner is, in fact, an experimental biochemist or geneticist who worked on fly, Drosophila. And his original idea was simply, when the first genome, or the more genes were known about Drosophila, the more people could do in the 80s and early 90s with Drosophila, the more he tried to sort of classify these proteins and built flybase. Some of you may have heard that. So whoever felt it was cold, I opened it a little bit, hoping that the induction of fresh oxygen will increase the happiness. The other way to increase the happiness is to say that soon it will be over. But anyway, so getting back to this story here.
That's the trick they use in casinos in Las Vegas. So people then get sleep and don't feel asleep, so they push more oxygen in the...
So you mean that I'm trying to sell you something and getting your maximum exposure time so that I win the most money? I believe you have a wrong perception of my job in this. Okay. The first realization was that Michael Ashburner had is you cannot do it in the sense you do it for the enzymes where you have one way of doing it that goes through. We need 3 different classes, 3 different ontologies. You have 3 different processes or 3 different phenomena, whatever you call it. Let's begin to call them. One is the biological process, called biological process. So this is about the pathway. This is about you're part of a group. So you're part of TUM, you're part of LMU, you're part of students, or you're part of student in Germany, you're part of students in biology and computer science, or these kind of groups you're part of, okay? And that is also the process. So it doesn't describe exactly who you are, it describes some of the background of what you do. And then there's molecular function. Molecular function is essentially this chemistry. And again, the issue of function that I talked about before, am I now talking about how the atom bond is changed? Or am I talking about this is part of the cell cycle? All of this is sort of part of molecular function. Okay? But it is something else than being part of a process. It is about the biochemistry in some sense of what this thing does. It is a structural protein, you said, versus it is an enzyme, for instance, right? That all would fit into this category of molecular function. And then there is something called cellular component in Go. And again, I will flash this by you many times, hoping that the more often you see it, the more likely you will remember some of it. And we will talk about localization. So essentially this is where in the cell are you? Where in the cell are you has to do with regulation, has to do with what you can interact with, has to do with what type of function you can actually access. Okay? Here is one of these hierarchies shown for DNA, a DNA binding protein, the process pathway, where again you see the sort of the hierarchy. Here is one shown for molecular function, nucleic acid binding, DNA binding, chromatin binding, and you get into more detail, and then you get the relations. And here's the localization, so essentially this is in the nucleus somewhere, in some particular part of the nucleus. And this is essentially exactly what I said before. Now, sequence determines structure, determines function. Sort of generic is the truth. Does that mean that a similar sequence has a similar structure? No, you say? Well, I would sort of debate the no on this one. What I show here is number of residues aligned, percentage sequence identity. Blue pairs are proteins that have similar structure by some criterion. Okay? So I could say the structure is different between 2 proteins if any atom is different. None of those is like that. They are defined such, there's some RMSD 2.5 angstrom, whatever it means. What it means is that if you look at the image, you may say they look alike. Okay? This is the level of similarity that we have. Recognizable similar. Okay? Not recognizable, oh, it's the same. But recognizable is, okay, there are some changes, but essentially it's the same. Right? This is the way the blue ones are done here. Every blue point here is a pair of proteins for which I know that they are, if you looked at the 3D, for all of those I know the 3D structure, if you looked at them you would say essentially they are the same structure, okay? The red ones, you would look at them and you say they're clearly not. There is something gray in between these two and I cut them out. So they're neither red nor blue. What you see is there is some function shown here. And the function is not such that everything above the line here is blue, because you see there's some red. But what you do see is this curve here is such that a lot of the blue is above, or a lot of what is above is blue. Put it like that. That means we have a simple function, is another metric that allows you from sequence to infer whether two proteins are likely to have a similar structure or not. Okay? So yes, similar sequences mostly have similar structure, and then there are exceptions. So your answer no was absolutely right. So here are extreme cases of no. So you were right, and the answer yes would have been right too. Most proteins with a very similar sequence have essentially similar structures, and then there are lots of exceptions, and lots of proteins that have similar structure have very different sequences. Okay, all of these things are true at the same time. That allows us comparative modeling, homology-based inference, and that allows us essentially to predict, in a short word, for half of all the protein sequences that we know, we can predict structure through that. Because there is something in the PDB, there is something in the database of known, experimentally known proteins that has a similar sequence, and we can infer structure. Okay. Now, does similar sequence in that same sense, again in a statistical sense, so there are many exceptions, but essentially you have a similar sequence, does it lead to a similar function? Same sequence essentially leads to the same function, but actually it's not that simple, because here we have an example where a protein with the same sequence in different tissues has different function. So we are already sort of seeing this clearly has the same structure, this protein, in these two different tissues. It has different functions because it has a slightly different environment. The different environment could be the proteins that it binds to in one tissue are not expressed in the other one. That would be one reason why this could happen. It could happen because it's actually in a different compartment. One is in the nucleus, one is secreted, and the protein looks slightly different because the pH of the environment is different. Again, we have examples for that. Now, then there are many proteins that are referred to as moonlighting proteins that essentially are like that in some sense even more, so that have two different functions. So essentially the idea of moonlighting comes from this idea that you have one job that you do during the day and one that you do at night. So this actually comes from New York. The doormen are people who moonlighten very often, meaning they have two jobs. They have one regular job during the day, and then they sit there and are doormen at night. This is where moonlighting comes from. And when you talk to them, they look like that. I'm just totally overloaded. Anyway, so there are a bunch of proteins. So this is, as you see here from the date, by now almost 20 years old. We know many of these moonlighting proteins. We still believe at this point they are exceptional. So yes, there are many, and here are some examples. Now comes the next issue. Does it mean that when I have a similar function, at least the proteins that do the same, or when I look at all the enzymes that do the same thing, do they have a similar sequence at least? And the answer is one example here, chymotrypsin and subtulin, very different structures, and this is now the kind of thing, you look at it and you really cannot recognize that there's similarity because they are totally different, and they do the same thing. There is one binding site that is the same. Those are the kinds of things where you can now ask, is that so convergent or divergent evolution? Did that happen by divergent evolution, or is that sort of a solution that was found from two different ways? And in many of these cases the discussion is not clear, yeah?
What do these proteins do?
I actually, chymotrypsin, I cannot answer your question. It cuts. Those are both enzymes. They both cut reactions. What the precise reaction is that they cut is, I don't know.
Sorry?
Yes?
But that different structure, like those proteins have different structures, but like they have the same function. But does it depend on the Substrate localization?
In this particular case, no.
No?
So they exist in the same substrate localization. It could be, but in this particular case, not. So there are enzymes that are done in the same type of cell that are part, in fact, even of the same pathway, of the same environment, and that do the same cutting or the same chemistry with the same active site, but with a very different sequence, with a different structure. So I just want to flash at you, there is this domain issue. Since proteins are made up of domains, most proteins have more than one structural unit. Typically we refer to that as a domain. A structural unit means that so the operational definition again is I cut that out, the red part here, and would adopt a unique three-dimensional structure on its own. Okay? That's one definition of a domain. And then now if you have in the database an annotation, here you have the ATP sulfurylase, here you have the the synthase, and you see that there is some sequence similarity. You have an unknown protein. Now you may call that ATP sulfurylase, but then, or this one, so you don't know in this annotation that you find in SwissProt whether this function that you see in the name of the protein relates to this domain, to that domain, or to the entire protein. Because most of the cases where you have, so most proteins in human are in fact, function is not annotated experimentally, and for the ones where it is, most of the cases you do not have exact local places where you say this function because there's the binding site. That's the exceptional case. When you want to, for instance, develop a method that predicts binding sites for enzymatic binding sites, and you want to have structural information. So let's look at some numbers. At this point, how many sequences do we know? Protein sequences. Any idea?
20,000.
200 million? We had this question.
The first part was good. The second part was not so good. 120 million. 120, so the reason why the second part was not so good is because 120. So SwissProt again, so what is the difference between SwissProt and UniProt, do you know? Yes. So the idea is SwissProt is curated. Is that what you said, curation?
Yeah.
So how do you guys imagine this curation? So again, the number is SwissProt has about 500,000 proteins, UniProt has 120 million. Okay? 500,000 are curated. What does it mean? Anybody? You all have seen that?
Yeah, there must have been some kind of
Evidence.
Yeah, evidence.
True.
Selection or something, and then.
Who does it? How do they do it? And where is that some kind of evidence? Is my question. Anybody?
I mean, you have manual curation, which is the highest quality but the most tedious. You also have some.
Okay, we have SwissProt. Is essentially most of the SwissProt is so the idea of SwissProt is manually curated. 500,000. So how do you imagine this? Is there one person sitting there for 2 years, or how do you imagine this? Do you have any idea what it takes to run SwissProt? I'm not talking about the computer science aspect of it. Manual curation. I lost your words. You said it's the most. What did you say? Most demanding? No, it's not what you said.
It's the most tedious.
Tedious is what you said. So most tedious translates to what? How do you imagine this operation? So again, let me tell you another thing before I get into that one. So essentially, if you plot SwissProt over time, you saw sort of some increase, and then you saw something like this at one particular point, where they removed about almost 20% or more than 20% of the SwissProt records, and much more than that in terms of the number of annotations. And the reason is because essentially they went through it in more detail and said that some of these annotations were not curated with high enough, or the curation was too optimistic, let's put it like that. Now, so there is the attempt to really have every record and every annotation in Swissprot be something that is verified by something else. Again, if you look at the annotations, I believe not even 1/5 of the annotations in Swissprot at the moment are really supported by experimental evidence. But Swissprot introduced evidence codes so you can distinguish one from the other. That's relatively new. But again, how do you imagine this operation? Any idea? What do you guys believe? How many people are involved? How long does it take? How do they do that? Anybody? And I'm going to stop the video because